Hello everyone, welcome back. Today I'm going to be talking about cell cycle, which is made up of interphase, mitosis, and cytokinesis. Okay, so every single cell in your body, in the human body, undergoes cell division. So there are actually two types that occur: mitosis, which occurs in the somatic or the body cells, which is the one I'm going to be talking about today. And then there's another one, which is meiosis. This only occurs in your gametes, which are yours, the sperm cells and the egg cells. But we're not going to be talking about that today. We're going to be talking about everything else outside of that. So basically, if it's not the egg cell, it's not a sperm cell, this is what we're going to be talking about. And this happens in three stages. The cell division happens in three stages: interphase, mitosis, and cytokinesis. And since we say that it's a cycle, we're going to see it, we have this little diagram here to show you that. So one of the first things you're going to see is that the green part, which is, this tells us that most of the cells spend their time in interphase. I'm going to be talking about the different subphases of that, which is G1. A lot of growth occurs there. Before a cell can divide, we basically need to double up everything. So like if you have like the 100 ribosomes in one cell, we need to duplicate that, because by the time we're done with the division, we basically are like trying to clone the cell. All right. And then the S phase, which is the second phase, here we're going to see basically DNA replicated. We're going to see DNA being made. And then the last phase is going to be G2 phase, which is just crossing the T's and dotting our I's and basically preparing the cell for cell division. And then the second stage would be mitosis, M phase for mitosis. And cytokinesis just kind of fits in there. It's not part of mitosis. Sometimes it looks like it's part of mitosis. It's still part of the M phase, but I'm going to be talking about that. So one of the things is we are going to start with one cell. All right, we're going to start here with one cell, and the end result will be two cells. This is how cells are able to multiply. However, first we have to talk about DNA replication. All right, so we need to replicate these DNA before we can, before the cell can undergo cell division. So DNA replication will first start with helicase enzymes. So helicases, if you remember when we were going over like the proteins, enzymes are going to end in ASE, right? So we are seeing here helicases. I have here helicases, DNA polymerase ligases. These are all types of enzymes. All right, I'll come back to this semi-conservative replication. So chromosome, you can see here chromosome, we unwind it and we kind of just zoom in and looking really at this. And just a quick recap, talking about DNA, they are made up of what we call bases, or I like to kind of like Lego blocks, and there are four bases for DNA: the adenine, all right, adenine, thymine, cytosine, and guanine. And adenine will always pair up with thymine, all right. It will always pair up with thymine, and cytosine will always pair up with guanine, and vice versa. So they would never switch places. They would never cross. If you have thymine, you can 100% guess this base is adenine is going to fit in there. If you have guanine and you have you're not sure what base, 100% sure cytosine is going to attach there. However, before we start replicating the DNA, we need to open it up, unzip it. So I put this picture of a zipper. We need to unzip it first. However, it's not just going to happen automatically. We need a certain enzyme, which is helicase, is going to unwind this the strands, because DNA is made up of a double helix, right? It's going to unwind it, and we're going to, by doing that, we are disrupting the hydrogen bonds. It's hydrogen bonds that basically hold these bases together. We're going to basically separate them. And as these strands unwind, the molecules of DNA polymerase are going to bind to them, all right? You're going to bind to them, and this will promote bonding between the bases, all right? This will allow, like I said, the adenine with the thymine, the cytosine with the guanine, and also linking all the nucleotides, all right? Another word for nucleotides are these bases. So DNA polymerase can work in only one direction. So someone might be like, wait, I see two arrows, one going in one direction, the other. They're going to be running in opposite directions, all right? So the two strands of the DNA molecules are oriented in opposite directions. The DNA polymerase are going to be bound to the upper strand, which is shown here. We're adding nucleotides to make a single continuous complementary copy, right? Where this is why, we have we're going to have a copy of the old, we're going to have a copy of the new, which I'm going to talk about when we come to semi-conservative replication, all right? And this grows towards the zipper. So we have what we will call like I said, the bubble has been open, one is going backwards, another is going forward. However, just because this is occurring doesn't mean like the bases are stable. I included here kind of like a stapler or a tape. This is where ligase comes in. Ligase has to make sure to splice the two segments together. And one of the things I want you to, by the way, this is a DNA polymerase. One of the things I want you to notice is that, we're saying replication, making a copy. If you go to, let's say, a printing store, you go to, Staples or Office Max and say, hey, I want to make a copy, right? You need to actually have like a paper. If you don't, they're like, do you want us to print something out so that you can make a copy of it, right? So this is a copy, and again, this ensures that what the cell, the DNA that we're being made, there's not going to be any error. I'm going to talk about transcription and translation later. There's not going to be any error. We're trying to basically clone it exactly the way it was before. If you shaved your eyebrows, the hair that's going to grow back should be exactly the one that was there before. You don't want like something purple hair growing, but this is due to DNA replication. And one of the things we're seeing here, for every new strand that we have a copy of the old and we have a copy of the new, all right? So in order to make a new copy, we need, a new DNA segment, we're going to have a new copy, which brings me to semi-conservative conservation. This is basically that for every DNA segment is going to have an old. Sometimes they also call it a parental strand, which is seen right here, a parental strand, and then this would be the new strand. So for every new copy, we're going to have a copy of the old, we're going to have a copy of the new. Another thing, I don't know if this is going to help, but the word liga from ligases means to tie together. So when all the nucleotides have taken their place, we need to basically staple them together, tie them together. All right, so eventually the unzipping will completely separate the original strands, and once the copying and last splicing are completed, you now have, like I said, two identical DNA molecules. Once the DNA has been replicated, the centrioles and enzymes and proteins have been made, the cell can then leave interphase, ready for mitosis, which I'm going to be talking about. So interphase, which is where most of these cells are going to spend their time, all right? And I mentioned briefly the three subphases, G1, S phase, and G2. I'll talk about the G0. So the G is short for gap, all right? Gap 1, this is where we see vigorous growth and metabolism of organelles. Every single organelle has to double up. If we have like 50 mitochondria, then we need to make that 100. If we have, I mentioned ribosomes, we have like 100 ribosomes, then we need to double that up. So all of that takes time. This is why it takes 8 to 12 hours, just this phase alone. And we also have checkpoints. I'm going to talk about checkpoints. Checkpoints is we just want to make sure that we do have the right amount before we are going to send the cell into mitosis, where division is going to take place. If we started off with 50 ribosomes, then we want to make sure we have 100, not 75, not 50. So making G1 checkpoint the most important checkpoint. And then G0, it's not really a phase. Cells that stop dividing are found in this phase. Muscle cells and neurons, they're stuck here. Why? Because, we'll talk about that when we get to the tissues. If a neuron gets damaged, it does not grow. It's not going to heal itself. I mean, we do have peripheral neurons. They have they have a chance of regenerating. It's very small, but when you compare it with other cells, it's not enough. However, your skin cells has the highest capacity for regeneration. So some cells, like muscle cells, like cardiac muscle cell, if some cardiac cells get damaged, then that's it. They never divide. So the next phase will be the S, or short for synthetic phase. This is where DNA replication occurs, all right? So we are going to duplicate chromosomes. This is going to take time. It's not a process we're going to rush because we don't want any genetic error. So it takes about 6 to 8 hours. And then the G2 phase, this is going to be where we're crossing our T's, dotting our I's. Does everything looks perfect? Because if not, we do not want a situation where, if something's off here, cancer might occur. All right. This takes 2 to 5 hours. This is just checking, double checking everything before they're they're being sent to mitosis. So the next phase is mitosis, and mitosis is basically for nuclear division. So we're going to see the nucleus dividing here. Mitosis has 4 subphases: prophase, metaphase, anaphase, and telophase. So prophase is the first phase. We're going to be talking about that. I kind of put an acronym to help you remember, PMAT, to know the order that they come in. Prophase, metaphase, anaphase. The word telo means end, like telomeres. Some of you have had bio background. Telomeres are ends, right? So that means to end something. All right, so let's take a look. So before we start looking at mitosis, I first want you to know what interphase looks like. So like I said, we're not going to be looking at the organelles because mitosis is really a nuclear division. So we're going to be concentrating on that. So it doesn't mean that this cell doesn't have organelles like mitochondria and rough ER and smooth ER, but we're not concerned with that right now because it's the job of mitosis. So one of the first things you're going to see, this is interphase right before mitosis occurs. We have centriosomes. There are going to be 2 of them, right? We double it. Usually, we're supposed to have 1 centriole per cell, but we have doubled everything, right? So this is why we have 2. All right, and then we're looking at the nucleus. You can see the nucleolus right here. This is the nuclear envelope. This is a chromatin. I want you to take a look at a chromatin. It looks like a bowl of spaghetti. All right, it's all jumbled up. There's no particular order here, right? See, so here we see chromosomes are visible, and another thing, as a result of the DNA replication, you start seeing the 2 copies of each chromosome now existing, and I cannot stress enough, chromatid. Chromatid is when you now have, they're basically like chromosomes, but they're not single. When they're single, we call them chromosomes. When they're in pairs, we call them chromatids. So that was early prophase. We're still in prophase, all right? Now we're in late prophase. So one of the things you're going to see, wow, look, the centrioles are at opposite ends of each other. And this nuclear envelope that we saw in early prophase looks like it's disappearing. It's disintegrating, all right? And we are seeing that the chromatids are being pulled apart by these microtubules, right? This mitotic spindle, they're being pulled apart, almost like, they're grabbing one end, one end of the pole is grabbing the chromatids, another end is grabbing them. And you are now able to really see the microtubules now. The second phase is metaphase. All right, metaphase. Another way you can remember this is midline. All the chromosomes are going to line up in the middle. All right, they're going to line up in the middle. We actually call this the metaphase plate. We say they line up across the equator. They line up in a single file. All right, a single file. So if you see a single file, you know that you're looking at metaphase. So anaphase is actually the shortest phase. A way to remember anaphase, think of A for apart. And I forgot to mention for metaphase, M for middle, M for midline. Anaphase, A for apart. Now these sister chromatids are being pulled apart. We don't call them chromatids anymore. Now we're calling them daughter chromosomes. This has nothing to do with gender. We don't have anything called male or son chromosomes. The term daughter is just a generic term for to let us know that they are now, due to the replication. They're now pulled apart. They're no longer in pairs. We see the centromere splitting, causing the chromatids to become chromosomes, and they're being pulled apart by kinetochores. The last phase is telophase. So telophase begins when the chromosome movement stops. The word telo, like I said, means end, like telomeres. So the two sets of chromosomes, we can see they start to uncoil to form chromatin. Chromatin is like a condensed, packed DNA. And you start seeing the nuclear envelope that disappeared in prophase. You start seeing it coming back. And you start seeing the nucleoli. The nucleoli starts to reappear as well. Something that the last time we saw this was in prophase. The spindle disappears. The spindle, those lines that seems to be pulling away the chromosomes, they disappear. However, we see the centrioles, all right? And someone might say, so why is cytokinesis? So cytokinesis actually begins during late anaphase, and it's almost happening at the same time that telophase is happening. And one unique thing about cytokinesis is you're going to see a cleavage furrow. All right, this cleavage furrow right here is kind of like, when there's a karate chop on a pillow. All right, so that's what this reminds me of. And this specific cleavage furrow is actually made up of actin microfilaments, which contracts to form this furrow. So we start seeing two distinct cells now, two daughter cells. They look exactly alike, identical, and each of them will have its own nucleus, which is identical to the original. Okay, so that concludes the topic of cell cycle. We've covered interphase, we've covered mitosis, and then we're ending it with cytokinesis. Okay, thank you.